Assalamu alaikum students. Students, hope so you all will be fine. Students, in this video, I am going to discuss about the important topic in chemistry of carbohydrates, and that is heteropolysaccharide. Okay students, it is a very important topic for short note, short answer question, and it is a very important topic for MCQ's point of view. So students, let's start our lecture. Heteropolysaccharide. Students, the another name for the heteropolysaccharide is mucopolysaccharide. Students, why mucopolysaccharides? Why mucopolysaccharide? Because heteropolysaccharide, it was first isolated from mucin, so that's why the name is given as mucopolysaccharide. So now coming toward the examples of mucopolysaccharide or heteropolysaccharide. Examples: the first one is glycosaminoglycan, second one is agar, third one is agarose, and the fourth one is pectin. Students, these are the examples of heteropolysaccharide or mucopolysaccharide: glycosaminoglycan, agar, agarose, and pectin. Students, today's the topic of our discussion is glycosaminoglycan. Today's topic of our discussion is glycosaminoglycan. This agar, this agar, and this agar, it is used for bacterial culture. This agar, it is used for bacterial culture, and agarose, this agarose, it is used for electrophoresis. Okay students, this agar and agarose, it is made up of glucose and galactose. And agar, it is used for bacterial culture. Agarose, it is used for the electrophoresis. Pectin, student, this pectin, it is used as a dietary fiber. Students, I am repeating again. Heteropolysaccharide, the another name is mucopolysaccharide. Why? Because heteropolysaccharide, it was first isolated from mucin, so that's why the name is given as mucopolysaccharide. The important examples of heteropolysaccharide or mucopolysaccharide is glycosaminoglycan, agar, agarose, and pectin. Today's topic of our discussion is glycosaminoglycan. This agar, it is used as a bacterial culture. Agarose, it is used for the electrophoresis, and pectin, it acts as a dietary fiber. So coming toward the glycosaminoglycan. Students, the glycosaminoglycan, It is made up of the repeating units of disaccharide. This glycosaminoglycan, it is made up of repeating disaccharide units. This glycosaminoglycan, it is made up of repeating disaccharide units, and these disaccharide units are amino sugar and uronic acid. The disaccharide units are amino sugar and uronic acid. Students, this amino sugar, it can be an acetyl glucosamine, or it can be an acetyl galactosamine. Okay, students. Students, uronic acid, it can be D-glucuronic acid or L-iduronic acid. Iduronic acid. This L-iduronic acid, basically, it is the epimer of this D-glucuronic acid. Students, I am repeating again. This glycosaminoglycan, it is made up of repeating disaccharide units, and these disaccharide units are amino sugar and uronic acid. This amino sugar, it can be an acetyl glucosamine and it can be an acetyl galactosamine. Uronic acid, it can be D-glucuronic acid and it can be L-iduronic acid. L-iduronic acid, it is the epimer of this D-glucuronic acid. Okay, students, now coming towards some important properties of glycosaminoglycan. Properties. Coming toward the some important properties of glycosaminoglycan. Students, this glycosaminoglycan, it contains large number of negatively charged groups that are carboxylate group, sulfate group. These are the negatively charged groups. Due to negatively charged groups, these glycosaminoglycan, they tend to repel each other due to same charges. So there are two important functions of the glycosaminoglycan are slippery slippery action slippery action of hyaluronic acid, and second one is they act as shock absorber. स्टूडेंट, शॉक एब्जॉर्बर का मतलब क्या है? शॉक एब्जॉर्बर इसको अंडरस्टैंड करने से पहले मैं आपको एक एग्जांपल से एक्सप्लेन करता हूँ. स्प्रिंग, when we squeeze the spring, it occupies a very small volume, and when we release it, it occupies a very large volume. So these glycosaminoglycan, they are also highly hydrated compound. In dehydrated form, or when we squeeze this glycosaminoglycan, then it occupies a small volume, and in hydrated form, when we release hyaluronic acid or glycosaminoglycan, they occupy a very large volume, and in this way they act as a shock absorber. I am repeating again. When we squeeze these glycosaminoglycan, they occupy a small volume, and when we release it, they occupy a large volume. Or we can say that in dehydrated form, because they are highly hydrated compound, in dehydrated form they occupy small volume, in hydrated form they occupy a large volume. Compressible property, we can say that this is a compressible property, and in this way they act as a shock absorber. And it is this such type of property, it is present in the synovial fluid of joints and vitreous humor of eye. Okay, students, glucose aminoglycan, the important component of glucose aminoglycan, that is a hyaluronic acid. There we will discuss now later the various glucose aminoglycan, and one of the most important glucose aminoglycan, that is hyaluronic acid. It is an important component of extracellular matrix. They are very important component of extracellular matrix. So students, the properties of glucose aminoglycan, this glucose aminoglycan contain large number of negatively charged groups, and due to this they occupy important function that are slippery action, and they act as a shock absorber, which is present in the synovial fluid of joints and vitreous humor of eye, and the important component of the extracellular matrix. Students, now coming toward the glucose aminoglycan. Take a screenshot, and then I will discuss the glucose aminoglycan. Okay. Glucose aminoglycan. The important examples of glucose aminoglycan. The important examples of glucose aminoglycan are hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin, heparin sulfate, and keratin sulfate. Students, these are the examples of the glucose aminoglycan. These are the examples of glucose aminoglycan: hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin, heparin sulfate, and keratin sulfate. Keratin sulfate. These are the examples of the glucose aminoglycan. Students, this this keratin sulfate, the important MCQs point of view मैं बताने जा रहा हूँ. It contains no uronic acid, ना ही इसमें डी ग्लूकुरोनिक एसिड होता है और ना ही इसमें एल यूरोनिक एसिड होता है. कोई भी यूरोनिक एसिड नहीं होता, और बल्कि इसमें it contains galactose, and galactose, it is the epimer of glucose. Okay, students, this dermatan sulfate, it contains L-iduronic acid, which is the epimer of the D-glucuronic acid. Okay, students, this chondroitin sulfate and dermatan sulfate, they contain N-acetyl galactosamine. N-acetyl N-acetylgalactosamine. Students, the examples of glycosaminoglycan are hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin sulfate, keratin sulfate. Keratin sulfate, it contains no uronic acid, it contains galactose, which is the epimer of glucose. This dermatan sulfate, L-iduronic acid, which is the epimer of D-glucuronic acid. This chondroitin sulfate and dermatan sulfate, they contain N-acetyl galactosamine. Okay, students, these are the important MCQs point of view that all glycosaminoglycan they contain D-glucuronic acid or uronic acid except this keratin sulfate, because keratin sulfate it contains galactose. It is not a uronic acid, it is the epimer of glucose. And also the another MCQ's point of view is that all glycosaminoglycan they contain D-glucuronic acid except this dermatan sulfate, because it contains L-iduronic acid, and L-iduronic acid is the epimer of D-glucuronic acid. स्टूडेंट्स, अब हम डिस्कस करेंगे इन ग्लाइकोसामिनोग्लिकन की कंपोजिशन को, इनके लोकेशन को और फंक्शंस को. सो लेट्स स्टार्ट. So first of all, hyaluronic acid. This hyaluronic acid, it is made up of N-acetyl glucosamine and D-glucuronic acid. Okay, students, and it is present in this hyaluronic acid is present in it is present in the synovial fluid of joints and vitreous humor of eye. This hyaluronic acid, it is present in the synovial fluid of joints and vitreous humor of eye. And the important functions of hyaluronic acid is they act as shock absorber, and they act as lubricant, and they are very important for cell migration, and in wound repair mechanism they are very important. Friends, these are the functions of the hyaluronic acid. This hyaluronic acid, it is made up of N-acetyl glucosamine and D-glucuronic acid. It is present in the synovial fluid of joints and vitreous humor of eye, and they act as shock absorber, they act as lubricant, they are responsible for cell migration, and they are very important in the wound repair mechanism. Now coming toward another glycosaminoglycan, that is chondroitin sulfate. This chondroitin sulfate, it is made up of N-acetyl galactosamine and D-glucuronic acid. This chondroitin sulfate, it is present in bone, cartilage and tendons. And this chondroitin sulfate, it is responsible for maintaining the maintaining shape and structure of bone, cartilage and tendon. Maintaining shape and structure of bone, cartilage and tendon. Now coming toward dermatan sulfate. It is made up of N-acetyl galactosamine and L-eduronic acid. L-eduronic acid is the epimer of the D-glucuronic acid. And this dermatan sulfate, it is present in skin, it is present in skin and heart valves. Students, the important function of dermatan sulfate is it is also responsible for maintaining shape and structure of skin and heart valves. Students, dermatan sulfate, it is responsible for plaque formation. So in this way, it is responsible for atherosclerosis. It is not function, and must remember this. Dermatan sulfate, it is responsible for plaque formation, and in this way they are very important in atherosclerosis. Students, another one is heparin. This heparin, it is made up of D-glucosamine, glucosamine and it also contains D-glucuronic acid and L-eduronic acid. Students, D-glucuronic acid and L-eduronic acid both are present in heparin. It is made up of D-glucosamine, D-glucosamine and D-glucuronic acid and as well as L-eduronic acid. Both uronic acid, they are present in heparin. And heparin, it is present in lungs, mast cells, blood and skin. And heparin, it acts as a anticoagulant. Okay, students, heparin, it is made up of D-glucosamine and D-glucuronic acid and L-eduronic acid. It is present in lungs, mast cells, blood and skin, and it is responsible for the anticoagulant. Students, next one is the heparin sulfate. heparin sulfate, मैं इसको थोड़ा रब कर रहा हूँ. heparin sulfate, heparin sulfate. Heparin sulfate, students, heparin sulfate is made up of D-glucosamine and D-glucuronic acid. Okay, and this heparin sulfate, it is present in the Bowman's capsule of glomerulus, and it is responsible for the permeability selective permeability of membrane. Okay, students, heparin sulfate, it is made up of D-glucosamine and D-glucuronic acid. Due to negatively charged groups, it is responsible for the selective permeability of membrane, and it is present in the Bowman's capsule of glomerulus. Next one is last one is keratin sulfate. Students, there are two types of keratin sulfate first and second. This keratin sulfate, it is made up of N-acetyl glucosamine and galactose. It contains no uronic acid, and the keratin sulfate first, it is present in cornea, and keratin sulfate second, it is present in cartilage. And the important functions of keratin sulfate is in cornea, it is responsible for the transparency of cornea. Transparency of cornea, and while in cartilage, it is responsible for maintaining the shape and structure of cartilage. Okay, students, keratin sulfate, there are two types, first and second. It is made up of N-acetyl glucosamine and galactose. And keratin sulfate first, it is present in cornea, while keratin sulfate second, it is present in cartilage. It is responsible for the transparency of cornea, and it is responsible for maintaining the shape and structure of cartilage. So students must remember the examples of glycosaminoglycan, that are hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin sulfate, and keratin sulfate. Keratin sulfate, it contains no uronic acid, it contains galactose, and the various are the hyaluronic acid and various examples, and as I already discussed in this lecture. So students, keep watching and thank you. It is a very important topic. Thank you.